The photo shows a hot spring with a vivid blue color in the middle and a golden color around the edge. Figure 22. Certain prokaryotes can live in extreme environments such as the Morning Glory Pool, a hot spring in Yellowstone National Park. The spring's vivid blue color is from the prokaryotes that thrive in its very hot waters. Credit: Modification of work by John Sullivan. Chapter outline: 22. 1. Prokaryotic diversity. 22. 2. Structure of prokaryotes, bacteria and archaea. 22. 3. Prokaryotic metabolism. 22. 4. Bacterial diseases in humans. 22. 5. Beneficial prokaryotes. In the recent past, scientists grouped living things into 6 kingdoms: Animalia, Plantae, Fungi, Protista, Archaea, and Bacteria, based on several criteria, such as the absence or presence of a nucleus and other membrane-bound organelles, the absence or presence of cell walls, multicellularity, and so on. In the late 20th century, the pioneering work of Carl Woese and others compared sequences of small subunit ribosomal RNA, SSU rRNA, which resulted in a more fundamental way to group organisms on Earth. Based on differences in the structure of cell membranes and in rRNA, Woese and his colleagues proposed that all life on Earth evolved along 3 lineages, called domains. The domain Bacteria comprises all organisms in the kingdom Bacteria, the domain Archaea comprises the rest of the prokaryotes, and the domain Eukarya comprises all eukaryotes, including organisms in the kingdoms Animalia, Plantae, Fungi, and Protista. 2 of the 3 domains, Bacteria and Archaea, are prokaryotic. Prokaryotes were the first inhabitants on Earth, appearing 3.5 to 3.8 billion years ago. These organisms are abundant and ubiquitous. That is, they are present everywhere. In addition to inhabiting moderate environments, they are found in extreme conditions, from boiling springs to permanently frozen environments in Antarctica, from salty environments like the Dead Sea to environments under tremendous pressure, such as the depths of the ocean, and from areas without oxygen, such as a waste management plant, to radioactively contaminated regions, such as Chernobyl. Prokaryotes reside in the human digestive system and on the skin, are responsible for certain illnesses, and serve an important role in the preparation of many foods. Learning objectives. By the end of this section, you will be able to do the following: describe the evolutionary history of prokaryotes, discuss the distinguishing features of extremophiles, explain why it is difficult to culture prokaryotes. Prokaryotes are ubiquitous. They cover every imaginable surface where there is sufficient moisture, and they also live on and inside virtually all other living things. In the typical human body, prokaryotic cells outnumber human body cells by about 10 to 1. They comprise the majority of living things in all ecosystems. Some prokaryotes thrive in environments that are inhospitable for most living things. Prokaryotes recycle nutrients, essential substances such as carbon and nitrogen, and they drive the evolution of new ecosystems, some of which are natural and others man-made. Prokaryotes have been on Earth since long before multicellular life appeared. Indeed, eukaryotic cells are thought to be the descendants of ancient prokaryotic communities. Prokaryotes, the first inhabitants of Earth. When and where did cellular life begin? What were the conditions on Earth when life began? We now know that prokaryotes were likely the first forms of cellular life on Earth, and they existed for billions of years before plants and animals appeared. The Earth and its moon are dated at about 454 billion years in age. This estimate is based on evidence from radiometric dating of meteorite material together with other substrate material from Earth and the moon. Early Earth had a very different atmosphere, contained less molecular oxygen than it does today, and was subjected to strong solar radiation. Thus, the first organisms probably would have flourished where they were more protected, such as in the deep ocean or far beneath the surface of the Earth. Strong volcanic activity was common on Earth at this time, so it is likely that these first organisms, the first prokaryotes, were adapted to very high temperatures. Because early Earth was prone to geological upheaval and volcanic eruption, and was subject to bombardment by mutagenic radiation from the sun, the first organisms were prokaryotes that must have withstood these harsh conditions. Microbial mats. Microbial mats, or large biofilms, may represent the earliest forms of prokaryotic life on Earth. There is fossil evidence of their presence starting about 3.5 billion years ago. It is remarkable that cellular life appeared on Earth only a billion years after the Earth itself formed, suggesting that precellular life that could replicate itself had evolved much earlier. A microbial mat is a multi-layered sheet of prokaryotes, figure 22. 2. That includes mostly bacteria, but also archaeans. Microbial mats are only a few centimeters thick, and they typically grow where different types of materials interface, mostly on moist surfaces. The various types of prokaryotes that comprise them carry out different metabolic pathways, and that is the reason for their various colors. Prokaryotes in a microbial mat are held together by a glue-like sticky substance that they secrete called extracellular matrix. The first microbial mats likely obtain their energy from chemicals found near hydrothermal vents. A hydrothermal vent is a breakage or fissure in the Earth's surface that releases geothermally heated water. With the evolution of photosynthesis about 3 billion years ago, some prokaryotes in microbial mats came to use a more widely available energy source, sunlight, whereas others were still dependent on chemicals from hydrothermal vents for energy and food. The part of photo shows a reddish-yellow mound with small chimneys growing out of it. Part B micrograph shows rod-shaped bacteria about 2 microns long swimming over a thicker mat of bacteria. Figure 22. 2. A microbial mat. A. This microbial mat, about 1 meter in diameter, is growing over a hydrothermal vent in the Pacific Ocean in a region known as the Pacific Ring of Fire. The mat's colony of bacteria helps retain microbial nutrients. Chimneys, such as the one indicated by the arrow, allow gases to escape. B. In this micrograph, bacteria are visualized using fluorescence microscopy. Credit a modification of work by Dr. Bob Embly, NOAAPMEL, Chief Scientist. Credit B. Modification of work by Ricardo Murga, Rodney Donlin, CDC. Scalebar data from Matt Russell. Stromatolites, fossilized microbial mats, represent the earliest record of life on Earth. A stromatolite is a sedimentary structure formed when minerals are precipitated out of water by prokaryotes in a microbial mat, figure 22. 3. Stromatolites form layered rocks made of carbonate or silicate. Although most stromatolites are artifacts from the past, there are places on Earth where stromatolites are still forming. For example, growing stromatolites have been found in the Anza-Borrego Desert State Park in San Diego County, California. Photo A shows a mass of gray mounds in shallow water. Photo B shows a swirl pattern in white and gray marbled rock. Figure 22, 3 stromatolites. A. These living stromatolites are located in Shark Bay, Australia. B. These fossilized stromatolites, found in Glacier National Park, Montana, are nearly 1.5 billion years old. Credit A. Robert Young, credit B. P. Carrara, NPS. The ancient atmosphere. Evidence indicates that during the first 2 billion years of Earth's existence, the atmosphere was anoxic, meaning that there was no molecular oxygen. Therefore, only those organisms that can grow without oxygen, anaerobic organisms, were able to live. Autotrophic organisms that convert solar energy into chemical energy are called phototrophs, and they appeared within 1 billion years of the formation of Earth. Then, cyanobacteria, also known as blue-green algae, evolved from these simple phototrophs at least 1 billion years later. It was the ancestral cyanobacteria, figure 22. 4. That began the oxygenation of the atmosphere. Increased atmospheric oxygen allowed the evolution of more efficient O2 utilizing catabolic pathways. It also opened up the land to increased colonization, because some O2 is converted into O3, ozone, and ozone effectively absorbs the ultraviolet light that could have otherwise caused lethal mutations in DNA. The current evidence suggests that the increase in O2 concentrations allowed the evolution of other life forms. This photo shows a woman squatting next to a stream of green-colored water. Figure 22. 4. Cyanobacteria. This hot spring in Yellowstone National Park flows toward the foreground. Cyanobacteria in the spring are green, and as water flows down the gradient, the intensity of the color increases as cell density increases. The water is cooler at the edges of the stream than in the center, causing the edges to appear greener. Credit Graciela Brels-Mariño. Microbes are adaptable, life in moderate and extreme environments. Some organisms have developed strategies that allow them to survive harsh conditions. Almost all prokaryotes have a cell wall, a protective structure that allows them to survive in both hypertonic and hypotonic aqueous conditions. Some soil bacteria are able to form endospores that resist heat and drought, thereby allowing the organism to survive until favorable conditions recur. These adaptations, along with others, allow bacteria to remain the most abundant life form in all terrestrial and aquatic ecosystems. Prokaryotes thrive in a vast array of environments. Some grow in conditions that would seem very normal to us, whereas others are able to thrive and grow under conditions that would kill a plant or an animal. Bacteria and archaea that are adapted to grow under extreme conditions are called extremophiles, meaning lovers of extremes. Extremophiles have been found in all kinds of environments. The depths of the oceans, hot springs, the Arctic and the Antarctic, in very dry places, deep inside Earth, in harsh chemical environments, and in high radiation environments. Figure 22. 5, just to mention a few. Because they have specialized adaptations that allow them to live in extreme conditions, many extremophiles cannot survive in moderate environments. There are many different groups of extremophiles. They are identified based on the conditions in which they grow best, and several habitats are extreme in multiple ways. For example, a soda lake is both salty and alkaline, so organisms that live in a soda lake must be both alkaliphiles and halophiles. Table 22. 1. Other extremophiles, like radioresistant organisms, do not prefer an extreme environment in this case, one with high levels of radiation, but have adapted to survive in it. Figure 22. 5. Organisms like these give us a better understanding of prokaryotic diversity and open up the possibility of finding new prokaryotic species that may lead to the discovery of new therapeutic drugs or have industrial applications. Extremophiles and their preferred conditions. Extremophile conditions for optimal growth. Acidophiles pH 3 or below. Alkaliphiles pH 9 or above. Thermophiles temperature 60-80 degrees C, 140-176 degrees F. Hyperthermophiles temperature 80-122 degrees C, 176-250 degrees F. Psychrophiles temperature of -15-10 degrees C, 550 degrees F or lower. Halophiles salt concentration of at least 0.2 m. Osmophiles high sugar concentration. Table 22. 1. This micrograph shows an oval Deinococcus cell about 2-5 microns in diameter, dividing. Figure 22. 5 radiation-tolerant prokaryotes. Deinococcus radiodurans, visualized in this false color transmission electron micrograph, is a prokaryote that can tolerate very high doses of ionizing radiation. It has developed DNA repair mechanisms that allow it to reconstruct its chromosome even if it has been broken into hundreds of pieces by radiation or heat. Credit: Modification of work by Michael Dolly. Scalebar data from Matt Russell. Prokaryotes in the Dead Sea. One example of a very harsh environment is the Dead Sea, a hypersaline basin that is located between Jordan and Israel. Hypersaline environments are essentially concentrated seawater. In the Dead Sea, the sodium concentration is 10 times higher than that of seawater, and the water contains high levels of magnesium, about 40 times higher than in seawater that would be toxic to most living things. Iron, calcium, and magnesium, elements that form divalent ions, Fe2+, Ca2+, and Mg2+, produce what is commonly referred to as hard water. Taken together, the high concentration of divalent cations, the acidic pH 60, and the intense solar radiation flux make the Dead Sea a unique and uniquely hostile ecosystem. 1. Figure 22. 6. What sort of prokaryotes do we find in the Dead Sea? The extremely salt-tolerant bacterial mats include Halobacterium, Halofrax vulcanae, which is found in other locations. Not only the Dead Sea, Halorubrum sodomens and Halobaculum gomorrens, and the Archaean Halorculum marismortui, among others. Photo A shows the Dead Sea and its accompanying brown shoreline. Micrograph B shows rod-shaped halobacteria. Figure 22, 6 halophilic prokaryotes. A. The Dead Sea is hypersaline. Nevertheless, salt-tolerant bacteria thrive in the sea. B. These halobacteria cells can form salt-tolerant bacterial mats. Credit A. Julian Menachini. Credit B. NASA. Scale-bar data from Matt Russell. Unculturable prokaryotes and the viable but non-culturable state. The process of culturing bacteria is complex and is one of the greatest discoveries of modern science. German physician Robert Koch is credited with discovering the techniques for pure culture, including staining and using growth media. Microbiologists typically grow prokaryotes in the laboratory using an appropriate culture medium containing all the nutrients needed by the target organism. The medium can be liquid, broth, or solid. After an incubation time at the right temperature, there should be evidence of microbial growth, figure 22. 7. Koch's assistant Julius Petri invented the Petri dish, whose use persists in today's laboratories. Koch worked primarily with the Mycobacterium tuberculosis bacterium that causes tuberculosis and developed guidelines, called Koch's postulates, to identify the organisms responsible for specific diseases. Koch's postulates continue to be widely used in the medical community. Koch's postulates include that an organism can be identified as the cause of disease when it is present in all infected samples and absent in all healthy samples, and it is able to reproduce the infection after being cultured multiple times. Today, cultures remain a primary diagnostic tool in medicine and other areas of molecular biology. 2 bacterial plates with red agar are shown. Both plates are covered with bacterial colonies. On the right plate, which contains hemolytic bacteria, the red agar has turned clear where bacteria are growing. On the left plate, which contains non-hemolytic bacteria, the agar is not clear. Figure 22. 7 bacteria growing on blood agar plates. In these agar plates, the growth medium is supplemented with red blood cells. Blood agar becomes transparent in the presence of hemolytic streptococcus, which destroys red blood cells and is used to diagnose streptococcus infections. The plate on the left is inoculated with non-hemolytic staphylococcus, large white colonies, and the plate on the right is inoculated with hemolytic streptococcus, tiny clear colonies. If you look closely at the right plate, you can see that the agar surrounding the bacteria has turned clear. Credit Bill Branson, NCI. Koch's postulates can be fully applied only to organisms that can be isolated and cultured. Some prokaryotes, however, cannot grow in a laboratory setting. In fact, over 99% of bacteria and archaea are unculturable. For the most part, this is due to a lack of knowledge as to what to feed these organisms and how to grow them. They may have special requirements for growth that remain unknown to scientists, such as needing specific micronutrients, pH, temperature, pressure, cofactors, or cometabolites. Some bacteria cannot be cultured because they are obligate intracellular parasites and cannot be grown outside a host cell. In other cases, culturable organisms become unculturable under stressful conditions, even though the same organism could be cultured previously. Those organisms that cannot be cultured but are not dead are in a viable but non-culturable VBNC state. The VBNC state occurs when prokaryotes respond to environmental stressors by entering a dormant state that allows their survival. The criteria for entering into the VBNC state are not completely understood. In a process called resuscitation, the prokaryote can go back to normal life when environmental conditions improve. Is the VBNC state an unusual way of living for prokaryotes? In fact, most of the prokaryotes living in the soil or in oceanic waters are non-culturable. It has been said that only a small fraction, perhaps 1%, of prokaryotes can be cultured under laboratory conditions. If these organisms are non-culturable, then how is it known whether they are present and alive? Microbiologists use molecular techniques, such as the polymerase chain reaction, PCR, to amplify selected portions of DNA of prokaryotes, e.g., 16 sRNA genes, demonstrating their existence. Recall that PCR can make billions of copies of a DNA segment in a process called amplification. The ecology of biofilms. Some prokaryotes may be unculturable because they require the presence of other prokaryotic species. Until a couple of decades ago, microbiologists used to think of prokaryotes as isolated entities living apart. This model, however, does not reflect the true ecology of prokaryotes, most of which prefer to live in communities where they can interact. As we have seen, a biofilm is a microbial community, figure 22. 8, held together in a gummy-textured matrix that consists primarily of polysaccharides secreted by the organisms, together with some proteins and nucleic acids. Biofilms typically grow attached to surfaces. Some of the best-studied biofilms are composed of prokaryotes, although fungal biofilms have also been described, as well as some composed of a mixture of fungi and bacteria. Biofilms are present almost everywhere. They can cause the clogging of pipes and readily colonize surfaces in industrial settings. In recent, large-scale outbreaks of bacterial contamination of food, biofilms have played a major role. They also colonize household surfaces, such as kitchen counters, cutting boards, sinks, and toilets, as well as places on the human body, such as the surfaces of our teeth. Interactions among the organisms that populate a biofilm, together with their protective exopolysaccharidic (EPS) environment, make these communities more robust than free-living or planktonic prokaryotes. The sticky substance that holds bacteria together also excludes most antibiotics and disinfectants, making biofilm bacteria hardier than their planktonic counterparts. Overall, biofilms are very difficult to destroy because they are resistant to many common forms of sterilization. Visual connection. During the first stage of biofilm development, a few bacteria adhere to a surface. During stage 2, the bacteria grow hairy appendages called pili. During stage 3, the microfilm grows into lumpy colonies. In stage 4, the microfilm grows into a more ball-like shape that is anchored to the surface by a smaller clump of bacteria. In stage 5, the ball of bacteria is larger, and bacteria with flagella swim away. Figure 22. 8 development of a biofilm. 5 stages of biofilm development are shown. During stage 1, initial attachment, bacteria adhere to a solid surface via weak van der Waals interactions, forces produced by induced electrical interactions between atoms. During stage 2, irreversible attachment, hair-like appendages called pili permanently anchor the bacteria to the surface. During stage 3, maturation I, the biofilm grows through cell division and recruitment of other bacteria. An extracellular matrix composed primarily of polysaccharides holds the biofilm together. During stage 4, maturation II, the biofilm continues to grow and takes on a more complex shape. During stage 5, dispersal, the biofilm matrix is partly broken down, allowing some bacteria to escape and colonize another surface. Micrographs of a Pseudomonas aeruginosa biofilm in each of the stages of development are shown. Credit D. Davis, Don Monroe, P. Loess. Compared to free-floating bacteria, bacteria in biofilms often show increased resistance to antibiotics and detergents. Why do you think this might be the case? Learning objectives. By the end of this section, you will be able to do the following. Describe the basic structure of a typical prokaryote. Describe important differences in structure between archaea and bacteria. There are many differences between prokaryotic and eukaryotic cells. The name prokaryote suggests that prokaryotes are defined by exclusion. They are not eukaryotes, or organisms whose cells contain a nucleus and other internal membrane-bound organelles. However, all cells have 4 common structures. The plasma membrane, which functions as a barrier for the cell and separates the cell from its environment. The cytoplasm, a complex solution of organic molecules and salts inside the cell. A double-stranded DNA genome, the informational archive of the cell, and ribosomes, where protein synthesis takes place. Prokaryotes come in various shapes, but many fall into 3 categories. Cocci spherical, bacilli rod-shaped, and spirili spiral-shaped. Figure 22. 9. Part A, the micrograph shows ball-shaped cocci about 800 nanometers across. Part B. The micrograph shows elongated, oval-shaped bacilli about 2,000 nanometers long. Part C. The micrograph shows corkscrew-shaped spirili that are quite long and 500 nanometers in diameter. Figure 22. 9 common prokaryotic cell types. Prokaryotes fall into 3 basic categories based on their shape, visualized here using scanning electron microscopy. A. Cocci or spherical, a pair is shown. B. Bacilli or rod-shaped, and C. Spirili or spiral-shaped. Credit, a modification of work by Janice Haney-Carr, Dr. Richard Faclam, CDC. Credit C. Modification of work by Dr. David Cox. Scale bar data from Matt Russell. The prokaryotic cell. Recall that prokaryotes are unicellular organisms that lack membrane-bound organelles or other internal membrane-bound structures. Figure 22. 10. Their chromosome, usually single, consists of a piece of circular, double-stranded DNA located in an area of the cell called the nucleoid. Most prokaryotes have a cell wall outside the plasma membrane. The cell wall functions as a protective layer, and it is responsible for the organism's shape. Some bacterial species have a capsule outside the cell wall. The capsule enables the organism to attach to surfaces, protects it from dehydration and attack by phagocytic cells, and makes pathogens more resistant to our immune responses. Some species also have flagella, singular, flagellum, used for locomotion, and pili, singular, pilus, used for attachment to surfaces including the surfaces of other cells. Plasmids, which consist of extrachromosomal DNA, are also present in many species of bacteria and archaea. In this illustration, the prokaryotic cell is rod-shaped. The circular chromosome is concentrated in a region called the nucleoid. The fluid inside the cell is called the cytoplasm. Ribosomes, depicted as small circles, float in the cytoplasm. The cytoplasm is encased by a plasma membrane, which in turn is encased by a cell wall. A capsule surrounds the cell wall. The bacterium depicted has a flagellum protruding from one narrow end. Pili are small protrusions that project from the capsule all over the bacterium, like hair. Figure 22. 10. The features of a typical prokaryotic cell. Flagella, capsules, and pili are not found in all prokaryotes. Recall that prokaryotes are divided into two different domains, bacteria and archaea, which together with eukarya comprise the three domains of life. Figure 22. 11. The trunk of the phylogenetic tree is a universal ancestor. The tree forms two branches. One branch leads to the domain bacteria, which includes the phyla Protobacteria, chlamydias, spirochetes, cyanobacteria, and gram-positive bacteria. The other branch branches again into the eukarya and archaea domains. Domain archaea includes the phyla Euryarchaeotes, Crenarchaeotes, Nanoarchaeotes, and Corarchaeotes. Figure 22. 11. The three domains of living organisms. Bacteria and archaea are both prokaryotes, but differ enough to be placed in separate domains. An ancestor of modern archaea is believed to have given rise to eukarya, the third domain of life. Major groups of archaea and bacteria are shown. Characteristics of bacterial phyla are described in Figure 22. 12 and Figure 22. 13. Major bacterial phyla include the Protobacteria, the chlamydias, the spirochetes, the photosynthetic cyanobacteria, and the gram-positive bacteria. The Protobacteria are in turn subdivided into several classes, from the alpha to the epsilon Protobacteria. Eukaryotic mitochondria are thought to be the descendants of alpha Protobacteria, while eukaryotic chloroplasts are derived from cyanobacteria. Archaeal phyla are described in Figure 22. 14. Characteristics of the 5 classes of bacterial phylum Protobacteria are described. The classes are alpha, beta, gamma, delta, and epsilon. Most species of alpha Protobacteria are photoautotrophic, but some are symbionts of plants and animals, and others are pathogens. Eukaryotic mitochondria are thought to be derived from bacteria in this group. Representative organisms include Rhizobium, a nitrogen-fixing endosymbiont associated with the roots of legumes, and Rickettsia, an obligate intracellular parasite that causes typhus and Rocky Mountain spotted fever, but not rickets, which is caused by vitamin D deficiency. A micrograph shows rod-shaped Rickettsia rickettsiae inside a much larger eukaryotic cell. Beta Protobacteria is a diverse group of bacteria. Some species play an important role in the nitrogen cycle. Representative organisms include nitrosomas, which oxidize ammonia into nitrite, and Spirillum minus, which causes rat bite fever. A micrograph of spiral-shaped Spirillum minus is shown. Gamma Protobacteria include many beneficial symbionts that populate the human gut, as well as familiar human pathogens. Some species from this subgroup oxidize sulfur compounds. Representative organisms include Escherichia coli, normally a beneficial microbe of the human gut, but some strains cause disease. Salmonella, certain strains of which cause food poisoning or typhoid fever. Yersinia pestis, the causative agent of bubonic plague. Pseudomonas aeruginosa, which causes lung infections. Vibrio cholera, the causative agent of cholera. And Chromatium, sulfur-producing bacteria that oxidize sulfur. Producing H2S micrograph shows rod-shaped Vibrio cholera, which are about 1 micron long. Some species of delta protobacteria generate a spore-forming fruiting body in adverse conditions. Others reduce sulfate and sulfur. Representative organisms include Myxobacteria, which generate spore-forming fruiting bodies in adverse conditions, and Desulfovibrio vulgaris, an anaerobic, sulfate-reducing bacterium. Micrograph shows a bent, rod-shaped Desulfovibrio vulgaris bacterium with a long flagellum. Epsilon protobacteria includes many species that inhabit the digestive tract of animals as symbionts or pathogens. Bacteria from this group have been found in deep-sea hydrothermal vents and cold seep habitats. Representative organisms include Campylobacter, which causes blood poisoning and intestinal inflammation, and Heliobacter pylori, which causes stomach ulcers. Micrograph shows short spiral or S-shaped Campylobacter bacteria. Figure 22, 12 the Protobacteria. Phylum Protobacteria is one of up to 52 bacteria phyla. Protobacteria is further subdivided into 5 classes, alpha through epsilon. Credit Rickettsia rickettsii. Modification of work by CDC. Credit Spirillum minus. Modification of work by Wolfram Adlisnig. Credit Vibrio cholera. Modification of work by Janus Haenikar, CDC. Credit Desulfovibrio vulgaris. Modification of work by Graham Bradley. Credit Campylobacter. Modification of work by DeWood, Pooley, USDA, ARS, EMU. Scalebar data from Matt Russell. This table describes the other 4 bacterial phyla classes. The first phylum described is Chlamydias. All members of this group are obligate intracellular parasites of animal cells. Cells' walls lack peptidoglycan. Micrograph shows a Pap smear of cells infected with Chlamydia trachomatis. Chlamydia infection is the most common sexually transmitted disease and can lead to blindness. All members of the phylum Spirochetes have spiral-shaped cells. Most are free-living anaerobes, but some are pathogenic. Flagella run lengthwise in the periplasmic space between the inner and outer membrane. Representative species include Treponema pallidum, the causative agent of syphilis and Borrelia burgdorferi. The causative agent of Lyme disease micrograph shows corkscrew-shaped Treponema pallidum, about 1 micron across. Bacteria in the phylum Cyanobacteria, also known as blue-green algae, obtain their energy through photosynthesis. They are ubiquitous, found in terrestrial, marine, and freshwater environments. Eukaryotic chloroplasts are thought to be derived from bacteria in this group. The Cyanobacterium prochlorococcus is believed to be the most abundant photosynthetic organism on Earth, responsible for generating half the world's oxygen. Micrograph shows a long, thin rod-shaped species called Formidium. Gram-positive bacteria have a thick cell wall and lack an outer membrane. Soil-dwelling members of this subgroup decompose organic matter. Some species cause disease. Representative species include Bacillus anthracis, which causes anthrax, Clostridium botulinum, which causes botulism, Clostridium difficile, which causes diarrhea during antibiotic therapy, Streptomyces, from which many antibiotics, including streptomycin, are derived, and Mycoplasmas, the smallest known bacteria, which lack a cell wall. Some are free-living, and some are pathogenic. Micrograph shows Clostridium difficile, which are rod-shaped and about 3 microns long. Figure 22, 13 other bacterial phyla. Chlamydia, Spirochetes, Cyanobacteria, and gram-positive bacteria are described in this table. Note that bacterial shape is not phylum-dependent. Bacteria within a phylum may be cocci, rod-shaped, or spiral. Credit Chlamydia trachomatis. Modification of work by Dr. Lance Liotta Laboratory, NCI. Credit Treponema pallidum. Modification of work by Dr. David Cox, CDC. Credit Formidium. Modification of work by USGS. Credit Clostridium difficile. Modification of work by Lois S. Wiggs, CDC. Scale bar data from Matt Russell. Characteristics of the 4 phyla of Archaea are described. Euryarchaeotes includes methanogens, which produce methane as a metabolic waste product, and halobacteria, which live in an extreme saline environment. Methanogens cause flatulence in humans and other animals. Halobacteria can grow in large blooms that appear reddish due to the presence of bacteriorhodopsin in the membrane. Bacteriorhodopsin is related to the retinal pigment rhodopsin. Micrograph shows rod-shaped halobacterium. Members of the ubiquitous Crenarchaeotes phylum play an important role in the fixation of carbon. Many members of this group are sulfur-dependent extremophiles. Some are thermophilic or hyperthermophilic. Micrograph shows cocci-shaped sulfolobus, a genus which grows in volcanic springs at temperatures between 75 degrees and 80 degrees Celsius and at a lowercase p uppercase h between 2 and 3. The phylum Nanoarchaeotes currently contains only one species, Nanoarchaeum equitans, which has been isolated from the bottom of the Atlantic Ocean and from the hydrothermal vent at Yellowstone National Park. It is an obligate symbiont with Ignicoccus, another species of Archaebacteria. Micrograph shows 2 small, round N-equitan cells attached to a larger Ignicoccus cell. Corarchaeotes are considered to be one of the most primitive forms of life, and so far have only been found in the Obsidian Pool, a hot spring at Yellowstone National Park. Micrograph shows a variety of specimens from this group which vary in shape. Figure 22. 14 archaeal phyla. Archaea are separated into 4 phyla: the Corarchaeota, Euryarchaeota, Crenarchaeota, and Nanoarchaeota. Credit Halobacterium. Modification of work by NASA. Credit Nanoarchaeotum equitans. Modification of work by Carl O. Stetter. Credit Corarchaeota. Modification of work by Office of Science of the U.S. Department of Energy. Scale bar data from Matt Russell. The plasma membrane of prokaryotes. The prokaryotic plasma membrane is a thin lipid bilayer, 6 to 8 nanometers, that completely surrounds the cell and separates the inside from the outside. Its selectively permeable nature keeps ions, proteins, and other molecules within the cell and prevents them from diffusing into the extracellular environment, while other molecules may move through the membrane. Recall that the general structure of a cell membrane is a phospholipid bilayer composed of 2 layers of lipid molecules. In archaeal cell membranes, isoprene, phytonyl chains linked to glycerol replace the fatty acids linked to glycerol in bacterial membranes. Some archaeal membranes are lipid monolayers instead of bilayers. Figure 22. 15. This illustration compares phospholipids from bacteria and eukarya to those from archaea. In bacteria and eukarya, fatty acids are attached to glycerol by an ester linkage, while in archaea, isoprene chains are linked to glycerol by an ether linkage. In the ester linkage, the first carbon in the fatty acid chain has an oxygen double bonded to it, whereas in the ether linkage, it does not. In archaea, the isoprene chains have methyl groups branching off from them, whereas such branches are absent in bacteria and eukarya. Both types of phospholipids result in similar lipid bilayers. Figure 22. 15 bacterial and archaeal phospholipids. Archaeal phospholipids differ from those found in bacteria and eukarya in 2 ways. First, they have branched phytonyl side chains instead of linear ones. Second, an ether bond instead of an ester bond connects the lipid to the glycerol. The cell wall of prokaryotes. The cytoplasm of prokaryotic cells has a high concentration of dissolved solutes. Therefore, the osmotic pressure within the cell is relatively high. The cell wall is a protective layer that surrounds some cells and gives them shape and rigidity. It is located outside the cell membrane and prevents osmotic lysis, bursting due to increasing volume. The chemical composition of the cell wall varies between archaea and bacteria, and also varies between bacterial species. Bacterial cell walls contain peptidoglycan, composed of polysaccharide chains that are cross-linked by unusual peptides containing both L and D amino acids, including D-glutamic acid and D-alanine. Proteins normally have only L amino acids. As a consequence, many of our antibiotics work by mimicking D amino acids and therefore have specific effects on bacterial cell wall development. There are more than 100 different forms of peptidoglycan. S-layer, surface layer, Proteins are also present on the outside of cell walls of both archaea and bacteria. Bacteria are divided into two major groups, gram-positive and gram-negative, based on their reaction to gram staining. Note that all gram-positive bacteria belong to two phyla, firmicutes and actinobacteria. Bacteria in the other phyla, protobacteria, chlamydias, spirochetes, cyanobacteria, and others, are gram-negative. The gram staining method is named after its inventor, Danish scientist Hans Christian Gram, 1853–1938. The different bacterial responses to the staining procedure are ultimately due to cell wall structure. Gram-positive organisms typically lack the outer membrane found in gram-negative organisms, figure 22. 16. Up to 90% of the cell wall in gram-positive bacteria is composed of peptidoglycan, and most of the rest is composed of acidic substances called teichoic acids. Teichoic acids may be covalently linked to lipids in the plasma membrane to form lipoteichoic acids. Lipoteichoic acids anchor the cell wall to the cell membrane. Gram-negative bacteria have a relatively thin cell wall composed of a few layers of peptidoglycan, only 10% of the total cell wall, surrounded by an outer envelope containing lipopolysaccharides, LPS, and lipoproteins. This outer envelope is sometimes referred to as a second lipid bilayer. The chemistry of this outer envelope is very different, however, from that of the typical lipid bilayer that forms plasma membranes. Visual connection. The left illustration shows the cell wall of gram-positive bacteria. The cell wall is a thick layer of peptidoglycan that exists outside the plasma membrane. A long, thin molecule called lipoteichoic acid anchors the cell wall to the cell membrane. The right illustration shows gram-negative bacteria. In gram-negative bacteria, a thin peptidoglycan cell wall is sandwiched between an outer and an inner plasma membrane. The space between the two membranes is called the periplasmic space. Lipoproteins anchor the cell wall to the outer membrane. Lipopolysaccharides protrude from the outer membrane. Porins are proteins in the outer membrane that allow entry of substances. Figure 22. 16. Cell walls in gram-positive and gram-negative bacteria. Bacteria are divided into two major groups, gram-positive and gram-negative. Both groups have a cell wall composed of peptidoglycan. In gram-positive bacteria, the wall is thick, whereas in gram-negative bacteria, the wall is thin. In gram-negative bacteria, the cell wall is surrounded by an outer membrane that contains lipopolysaccharides and lipoproteins. Porins are proteins in the cell membrane that allow substances to pass through the outer membrane of gram-negative bacteria. In gram-positive bacteria, lipoteichoic acid anchors the cell wall to the cell membrane. Credit: modification of work by Francis Cosp 2/Wikimedia Commons. Which of the following statements is true? Gram-positive bacteria have a single cell wall anchored to the cell membrane by lipoteichoic acid. Porins allow entry of substances into both gram-positive and gram-negative bacteria. The cell wall of gram-negative bacteria is thick, and the cell wall of gram-positive bacteria is thin. Gram-negative bacteria have a cell wall made of peptidoglycan, whereas gram-positive bacteria have a cell wall made of lipoteichoic acid. Archaean cell walls do not have peptidoglycan. There are 4 different types of archaean cell walls. One type is composed of pseudopeptidoglycan, which is similar to peptidoglycan in morphology, but contains different sugars in the polysaccharide chain. The other 3 types of cell walls are composed of polysaccharides, glycoproteins, or pure protein. Other differences between bacteria and archaea are seen in Table 22. 2. Note that features related to DNA replication, transcription, and translation in archaea are similar to those seen in eukaryotes. Differences and similarities between bacteria and archaea. Structural characteristic bacteria archaea. Cell type prokaryotic. Cell morphology variable. Cell wall contains peptidoglycan, does not contain peptidoglycan. Cell membrane type lipid bilayer, or lipid monolayer. Plasma membrane lipids, fatty acids, glycerol ester, phytonylglycerol ethers. Chromosome typically circular. Replication origins single multiple. RNA polymerase single multiple. Initiator tRNA formal methionine, streptomycin inhibition, sensitive resistant. Calvin cycle yes no. Table 22. 2. Reproduction. Reproduction in prokaryotes is asexual and usually takes place by binary fission. Recall that the DNA of a prokaryote is a single, circular chromosome. Prokaryotes do not undergo mitosis. Instead, the chromosome is replicated and the two resulting copies separate from one another due to the growth of the cell. The prokaryote, now enlarged, is pinched inward at its equator and the two resulting cells, which are clones, separate. Binary fission does not provide an opportunity for genetic recombination or genetic diversity, but prokaryotes can share genes by three other mechanisms. In transformation, the prokaryote takes in DNA shed by other prokaryotes into its environment. If a non-pathogenic bacterium takes up DNA for a toxin gene from a pathogen and incorporates the new DNA into its own chromosome, it too may become pathogenic. In transduction, bacteriophages, the viruses that infect bacteria, may move short pieces of chromosomal DNA from one bacterium to another. Transduction results in a recombinant organism. Archaea also have viruses that may translocate genetic material from one individual to another. In conjugation, DNA is transferred from one prokaryote to another by means of a pilus, which brings the organisms into contact with one another and provides a channel for transfer of DNA. The DNA transferred can be in the form of a plasmid or as a composite molecule, containing both plasmid and chromosomal DNA. These three processes of DNA exchange are shown in Figure 22. 17. Reproduction can be very rapid, a few minutes for some species. This short generation time, coupled with mechanisms of genetic recombination and high rates of mutation, result in the rapid evolution of prokaryotes, allowing them to respond to environmental changes, such as the introduction of an antibiotic, very quickly. Illustration A shows transformation, where a small, circular piece of DNA is absorbed by a prokaryotic cell. The DNA then gets incorporated in the genome. Illustration B shows transduction, where a bacteriophage injects DNA into a prokaryotic cell. The DNA then gets incorporated in the genome. Illustration C shows conjugation, where two bacteria are connected by a pilus. A small loop of DNA is transferred from one cell to the other via the pilus. Figure 22. 17. Gene transfer mechanisms in prokaryotes. There are three mechanisms by which prokaryotes can exchange DNA. In a transformation, the cell takes up prokaryotic DNA directly from the environment. The DNA may remain separate as plasmid DNA or be incorporated into the host genome. In B transduction, a bacteriophage injects DNA into the cell that contains a small fragment of DNA from a different prokaryote. In C conjugation, DNA is transferred from one cell to another via a mating bridge, or pilus, that connects the two cells after the sex pilus draws the two bacteria close enough to form the bridge. Evolution connection. The evolution of prokaryotes. How do scientists answer questions about the evolution of prokaryotes? Unlike with animals, artifacts in the fossil record of prokaryotes offer very little information. Fossils of ancient prokaryotes look like tiny bubbles in rock. Some scientists turn to genetics and to the principle of the molecular clock, which holds that the more recently two species have diverged, the more similar their genes, and thus proteins, will be. Conversely, species that diverged long ago will have more genes that are dissimilar. Scientists at the NASA Astrobiology Institute and at the European Molecular Biology Laboratory collaborated to analyze the molecular evolution of 32 specific proteins common to 72 species of prokaryotes. 2. The model they derived from their data indicates that three important groups of bacteria, Actinobacteria, Deinococcus, and Cyanobacteria, collectively called Pterobacteria by the authors, were the first to colonize land. Actinobacteria are a group of very common gram-positive bacteria that produce branch structures like fungal mycelia, and include species important in decomposition of organic wastes. You will recall that Deinococcus is a genus of bacterium that is highly resistant to ionizing radiation. It has a thick peptidoglycan layer in addition to a second external membrane, so it has features of both gram-positive and gram-negative bacteria. Cyanobacteria are photosynthesizers and were probably responsible for the production of oxygen on the ancient Earth. The timelines of divergence suggest that bacteria, members of the domain Bacteria, diverged from common ancestral species between 2.5 and 3.2 billion years ago, whereas the Archaea diverged earlier, between 3.1 and 4.1 billion years ago. Eukarya later diverged from the Archean line. The work further suggests that stromatolites that formed prior to the advent of cyanobacteria, about 2.6 billion years ago, photosynthesized in an anoxic environment, and that because of the modifications of the pterobacteria for land, resistance to drying, and the possession of compounds that protect the organism from excess light. Photosynthesis using oxygen may be closely linked to adaptations to survive on land. Learning objectives. By the end of this section, you will be able to do the following. Identify the macronutrients needed by prokaryotes and explain their importance. Describe the ways in which prokaryotes get energy and carbon for life processes. Describe the roles of prokaryotes in the carbon and nitrogen cycles. Prokaryotes are metabolically diverse organisms. In many cases, a prokaryote may be placed into a species clade by its defining metabolic features. Can it metabolize lactose? Can it grow on citrate? Does it produce H2S? Does it ferment carbohydrates to produce acid and gas? Can it grow under anaerobic conditions? Since metabolism and metabolites are the product of enzyme pathways, and enzymes are encoded in genes, the metabolic capabilities of a prokaryote are a reflection of its genome. There are many different environments on Earth with various energy and carbon sources, and variable conditions to which prokaryotes may be able to adapt. Prokaryotes have been able to live in every environment from deep-water volcanic vents to Antarctic ice by using whatever energy and carbon sources are available. Prokaryotes fill many niches on Earth, including involvement in nitrogen and carbon cycles, photosynthetic production of oxygen, decomposition of dead organisms, and thriving as parasitic, commensal, or mutualistic organisms inside multicellular organisms, including humans. The very broad range of environments that prokaryotes occupy is possible because they have diverse metabolic processes. Needs of prokaryotes. The diverse environments and ecosystems on Earth have a wide range of conditions in terms of temperature, available nutrients, acidity, salinity, oxygen availability, and energy sources. Prokaryotes are very well equipped to make their living out of a vast array of nutrients and environmental conditions. To live, prokaryotes need a source of energy, a source of carbon, and some additional nutrients. Macronutrients. Cells are essentially a well-organized assemblage of macromolecules and water. Recall that macromolecules are produced by the polymerization of smaller units called monomers. For cells to build all of the molecules required to sustain life, they need certain substances, collectively called nutrients. When prokaryotes grow in nature, they must obtain their nutrients from the environment. Nutrients that are required in large amounts are called macronutrients, whereas those required in smaller or trace amounts are called micronutrients. Just a handful of elements are considered macronutrients: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. A mnemonic for remembering these elements is the acronym CHONPS. Why are these macronutrients needed in large amounts? They are the components of organic compounds in cells, including water. Carbon is the major element in all macromolecules. Carbohydrates, proteins, nucleic acids, lipids, and many other compounds. Carbon accounts for about 50% of the composition of the cell. In contrast, nitrogen represents only 12% of the total dry weight of a typical cell. Nitrogen is a component of proteins, nucleic acids, and other cell constituents. Most of the nitrogen available in nature is either atmospheric nitrogen, N2, or another inorganic form. Diatomic N2 nitrogen, however, can be converted into an organic form only by certain microorganisms, called nitrogen-fixing organisms. Both hydrogen and oxygen are part of many organic compounds and of water. Phosphorus is required by all organisms for the synthesis of nucleotides and phospholipids. Sulfur is part of the structure of some amino acids such as cysteine and methionine, and is also present in several vitamins and coenzymes. Other important macronutrients are potassium, K, magnesium, Mg, calcium, Ca, and sodium, Na. Although these elements are required in smaller amounts, they are very important for the structure and function of the prokaryotic cell. Micronutrients. In addition to these macronutrients, prokaryotes require various metallic elements in small amounts. These are referred to as micronutrients or trace elements. For example, iron is necessary for the function of the cytochromes involved in electron transport reactions. Some prokaryotes require other elements, such as boron B, chromium Cr, and manganese, Mn, primarily as enzyme cofactors. The ways in which prokaryotes obtain energy. Prokaryotes are classified both by the way they obtain energy and by the carbon source they use for producing organic molecules. These categories are summarized in Table 22. 3 prokaryotes can use different sources of energy to generate the ATP needed for biosynthesis and other cellular activities. Phototrophs or phototrophic organisms obtain their energy from sunlight. Phototrophs trap the energy of light using chlorophylls or, in a few cases, bacterial rhodopsin. Rhodopsin-using phototrophs, oddly, are phototrophic, but not photosynthetic, since they do not fix carbon. Chemotrophs, or chemosynthetic organisms, obtain their energy from chemical compounds. Chemotrophs that can use organic compounds as energy sources are called chemoorganotrophs. Those that can use inorganic compounds, like sulfur or iron compounds, as energy sources are called chemolithotrophs. Energy-producing pathways may be either aerobic, using oxygen as the terminal electron acceptor, or anaerobic, using either simple inorganic compounds or organic molecules as the terminal electron acceptor. Since prokaryotes lived on Earth for nearly 1 billion years before photosynthesis produced significant amounts of oxygen for aerobic respiration, many species of both bacteria and archaea are anaerobic, and their metabolic activities are important in the carbon and nitrogen cycles discussed below. The ways in which prokaryotes obtain carbon. Prokaryotes not only can use different sources of energy, but also different sources of carbon compounds. Autotrophic prokaryotes synthesize organic molecules from carbon dioxide. In contrast, heterotrophic prokaryotes obtain carbon from organic compounds. To make the picture more complex, the terms that describe how prokaryotes obtain energy and carbon can be combined. Thus, photoautotrophs use energy from sunlight and carbon from carbon dioxide and water, whereas chemoheterotrophs obtain both energy and carbon from an organic chemical source. Chemolithoeutrophs obtain their energy from inorganic compounds, and they build their complex molecules from carbon dioxide. Finally, prokaryotes that get their energy from light, but their carbon from organic compounds, are photoheterotrophs. The table below, Table 22. 3, summarizes carbon and energy sources in prokaryotes. Carbon and energy sources in prokaryotes. Energy source, electron source, carbon source, nutritional type. Light, phototroph, organic material. Organotroph, organic material. Heterotroph, photoorganoheterotroph. Carbon dioxide. Autotroph, inorganic material. Lithotroph, organic material. Heterotroph, carbon dioxide. Autotroph, photolithotroph. Chemicals, chemotroph, organic material. Organotroph, organic material. Heterotroph, chemoorganoheterotroph. Carbon dioxide. Autotroph, inorganic material. Lithotroph, organic material. Heterotroph, chemolithoheterotroph. Carbon dioxide. Autotroph, chemolithoeutotroph. Table 22. 3. Role of prokaryotes in ecosystems. Prokaryotes are ubiquitous. There is no niche or ecosystem in which they are not present. Prokaryotes play many roles in the environments they occupy. The roles they play in the carbon and nitrogen cycles are vital to life on Earth. In addition, the current scientific consensus suggests that metabolically interactive prokaryotic communities may have been the basis for the emergence of eukaryotic cells. Prokaryotes and the carbon cycle. Carbon is one of the most important macronutrients, and prokaryotes play an important role in the carbon cycle, Figure 22. 18. The carbon cycle traces the movement of carbon from inorganic to organic compounds and back again. Carbon is cycled through Earth's major reservoirs: land, the atmosphere, aquatic environments, sediments and rocks, and biomass. In a way, the carbon cycle echoes the role of the 4 elements: first proposed by the ancient Greek philosopher, Empedocles: fire, water, earth, and air. Carbon dioxide is removed from the atmosphere by land plants and marine prokaryotes, and is returned to the atmosphere via the respiration of chemoorganotrophic organisms, including prokaryotes, fungi, and animals. Although the largest carbon reservoir in terrestrial ecosystems is in rocks and sediments, that carbon is not readily available. Participants in the carbon cycle are roughly divided among producers, consumers, and decomposers of organic carbon compounds. The primary producers of organic carbon compounds from CO2 are land plants and photosynthetic bacteria. A large amount of available carbon is found in living land plants. A related source of carbon compounds is humus, which is a mixture of organic materials from dead plants and prokaryotes that have resisted decomposition. The term humus, by the way, is the root of the word human. Consumers such as animals and other heterotrophs use organic compounds generated by producers and release carbon dioxide to the atmosphere. Other bacteria and fungi, collectively called decomposers, carry out the breakdown, decomposition of plants and animals and their organic compounds. Most carbon dioxide in the atmosphere is derived from the respiration of microorganisms that decompose dead animals, plants, and humus. In aqueous environments and their anoxic sediments, there is another carbon cycle taking place. In this case, the cycle is based on one-carbon compounds. In anoxic sediments, prokaryotes, mostly Archaea, produce methane, CH4. This methane moves into the zone above the sediment, which is richer in oxygen and supports bacteria called methane oxidizers that oxidize methane to carbon dioxide, which then returns to the atmosphere. An illustration shows the carbon life cycle. Carbon dioxide in the atmosphere occurs by way of volcanic eruptions, animal, microbial, and marine respiration, and human emissions, such as from factories. It is absorbed by terrestrial photosynthesis from plants and marine photosynthesis. The weathering of terrestrial rocks turns into soil carbon, which then branches into either fossil carbon or directly to microbial respiration and decomposition. Fossil carbon also contributes to microbial respiration and decomposition. This decomposition has a leaching or runoff effect and enters the ocean as sediments. Those sediments can be uplifted and also end up contributing to microbial respiration and decomposition. Figure 22. 18. The carbon cycle. Prokaryotes play a significant role in continuously moving carbon through the biosphere. Credit, modification of work by John M. Evans and Howard Perlman, USGS. Prokaryotes and the nitrogen cycle. Nitrogen is a very important element for life because it is a major constituent of proteins and nucleic acids. It is a macronutrient, and in nature, it is recycled from organic compounds to ammonia, ammonium ions, nitrate, nitrite, and nitrogen gas by many processes, many of which are carried out only by prokaryotes, as illustrated in Figure 22. 19. Prokaryotes are key to the nitrogen cycle. The largest pool of nitrogen available in the terrestrial ecosystem is gaseous nitrogen, N2 from the air, but this nitrogen is not usable by plants, which are primary producers. Gaseous nitrogen is transformed or fixed into more readily available forms, such as ammonia, NH3, through the process of nitrogen fixation. Nitrogen-fixing bacteria include Azotobacter in soil and the ubiquitous photosynthetic cyanobacteria. Some nitrogen-fixing bacteria, like Rhizobium, live in symbiotic relationships in the roots of legumes. Another source of ammonia is aminification, the process by which ammonia is released during the decomposition of nitrogen-containing organic compounds. The ammonium ion is progressively oxidized by different species of bacteria in a process called nitrification. The nitrification process begins with the conversion of ammonium to nitrite, NO2, and continues with the conversion of nitrite to nitrate. Nitrification in soils is carried out by bacteria belonging to the genera Nitrosomas, Nitrobacter, and Nitrospira. Most nitrogen in soil is in the form of ammonium, NH4+, or nitrate, NO3. Ammonia and nitrate can be used by plants or converted to other forms. Ammonia released into the atmosphere, however, represents only 15% of the total nitrogen released. The rest is as N2 and N2O, nitrous oxide. Ammonia is catabolized anaerobically by some prokaryotes, yielding N2 as the final product. Denitrifying bacteria reverse the process of nitrification, reducing the nitrate from soils to gaseous compounds such as N2O, NO, and N2. Visual connection. This illustration shows the role of bacteria in the nitrogen cycle. Nitrogen-fixing bacteria in root nodules of legumes convert nitrogen gas, or uppercase N2, into organic nitrogen found in plants. Nitrogen-fixing soil bacteria produce ammonium ion, or upper N upper H4+ sign. Decomposers, including bacteria and fungi, decompose organic matter, also releasing upper N upper H4+ sign. Nitrification is the process by which nitrifying bacteria produce nitrites, shown as upper N upper O2 negative, and nitrates, shown as upper N upper O3 negative. Nitrates are assimilated by plants, then animals, then decomposers. Denitrifying bacteria convert nitrates to nitrogen gas, completing the cycle. Figure 22:19 The nitrogen cycle. Prokaryotes play a key role in the nitrogen cycle. Credit, Environmental Protection Agency. Which of the following statements about the nitrogen cycle is false? Nitrogen-fixing bacteria exist on the root nodules of legumes and in the soil. Denitrifying bacteria convert nitrates, NO3, into nitrogen gas, N2. Ammonification is the process by which ammonium ion, NH4+, is released from decomposing organic compounds. Nitrification is the process by which nitrites, NO2, are converted to ammonium ion, NH4+. Learning objectives. By the end of this section, you will be able to do the following. Identify bacterial diseases that caused historically important plagues and epidemics. Describe the link between biofilms and foodborne diseases. Explain how overuse of antibiotics may be creating superbugs. Explain the importance of MRSA with respect to the problems of antibiotic resistance. To a prokaryote, humans may be just another housing opportunity. Unfortunately, the tendency of some species can have harmful effects and cause disease. Bacteria or other infectious agents that cause harm to their human hosts are called pathogens. Devastating pathogen-borne diseases and plagues, both viral and bacterial in nature, have affected humans and their ancestors for millions of years. The true cause of these diseases was not understood until modern scientific thought developed, and many people thought that diseases were a spiritual punishment. Only within the past several centuries have people understood that staying away from afflicted persons, disposing of the corpses and personal belongings of victims of illness, and sanitation practices reduced their own chances of getting sick. Epidemiologists study how diseases are transmitted and how they affect a population. Often, they must following the course of an epidemic, a disease that occurs in an unusually high number of individuals in a population at the same time. In contrast, a pandemic is a widespread and usually worldwide epidemic. An endemic disease is a disease that is always present, usually at low incidence, in a population. Long history of bacterial disease. There are records about infectious diseases as far back as 3000 B.C. A number of significant pandemics caused by bacteria have been documented over several hundred years. Some of the most memorable pandemics led to the decline of cities and entire nations. In the 21st century, infectious diseases remain among the leading causes of death worldwide, despite advances made in medical research and treatments in recent decades. A disease spreads when the pathogen that causes it is passed from one person to another. For a pathogen to cause disease, it must be able to reproduce in the host's body and damage the host in some way. The Plague of Athens. In 430 BC, the Plague of Athens killed 1/4 of the Athenian troops who were fighting in the Great Peloponnesian War and weakened Athens's dominance and power. The plague impacted people living in overcrowded Athens, as well as troops aboard ships that had to return to Athens. The source of the plague may have been identified recently when researchers from the University of Athens were able to use DNA from teeth recovered from a mass grave. The scientists identified nucleotide sequences from a pathogenic bacterium, Salmonella enterica serovar typhi, figure 22:20, which causes typhoid fever. 3. This disease is commonly seen in overcrowded areas and has caused epidemics throughout recorded history. Micrograph shows pink rod-shaped bacteria. Figure 22. 20 Salmonella enterica. Salmonella enterica cerevar typhi, the causative agent of typhoid fever, is a gram-negative, rod-shaped gamma protobacterium. Typhoid fever, which is spread through feces, causes intestinal hemorrhage, high fever, delirium, and dehydration. Today, between 16 and 33 million cases of this re-emerging disease occur annually, resulting in over 200,000 deaths. Carriers of the disease can be asymptomatic. In a famous case in the early 1900s, a cook named Mary Mallon, Typhoid Mary, unknowingly spread the disease to over 50 people, 3 of whom died. Other serotypes of Salmonella cause food poisoning. Credit: modification of work by NCI, CDC. Bubonic plagues. From 541 to 750, the plague of Justinian, an outbreak of what was likely bubonic plague, eliminated 1/4 to 1/2 of the human population in the eastern Mediterranean region. The population in Europe dropped by 50% during this outbreak. Astoundingly, bubonic plague would strike Europe more than once. Bubonic plague is caused by the bacterium Yersinia pestis. One of the most devastating pandemics attributed to bubonic plague was the Black Death, 1346 to 1361. It is thought to have originated in China and spread along the Silk Road, a network of land and sea trade routes, to the Mediterranean region and Europe, carried by fleas living on black rats that were always present on ships. The Black Death was probably named for the tissue necrosis, figure 22, 21c, that can be one of the symptoms. The buboes of bubonic plague were painfully swollen areas of lymphatic tissue, a pneumonic form of the plague, spread by the coughing and sneezing of infected individuals, spreads directly from human to human and can cause death within a week. The pneumonic form was responsible for the rapid spread of the Black Death in Europe. The Black Death reduced the world's population from an estimated 450 million to about 350 to 375 million. Bubonic plague struck London yet again in the mid-1600s, figure 22. 21. In modern times, approximately 1,000 to 3,000 cases of plague arise globally each year, and a sylvatic form of plague, carried by fleas living on rodents such as prairie dogs and black-footed ferrets, infects 10 to 20 people annually in the American Southwest. Although contracting bubonic plague before antibiotics meant almost certain death, the bacterium responds to several types of modern antibiotics, and mortality rates from plague are now very low. Illustration A shows 2 men loading a dead body onto a cart. Another body lies in the street. Label beneath the illustration says, Plague in 1665. Micrograph B shows rod-shaped bacteria. Photo C shows a man with black gangrene on his fingers, arm, nose, and lips. Figure 22. 21. The Black Death. The Great Plague of London killed an estimated 200,000 people, or about 20% of the city's population. The causative agent, the B. bacterium Yersinia pestis, is a gram-negative, rod-shaped bacterium from the class Gamma protobacteria. The disease is transmitted through the bite of an infected flea, which is carried on a rodent. Symptoms include swollen lymph nodes, fever, seizure, vomiting of blood, and C. gangrene. Credit B. Rocky Mountain Laboratories, NIAID, NIH, scale bar data from Matt Russell, credit C. Textbook of Military Medicine, Washington, D.C., U.S. Department of the Army, Office of the Surgeon General, Borden Institute. Link to learning. Watch a video on the modern understanding of the Black Death, bubonic plague in Europe during the 14th century. Migration of diseases to new populations. One of the negative consequences of human exploration was the accidental biological warfare that resulted from the transport of a pathogen into a population that had not previously been exposed to it. Over the centuries, Europeans tended to develop genetic immunity to endemic infectious diseases. But when European conquerors reached the Western Hemisphere, they brought with them disease-causing bacteria and viruses, which triggered epidemics that completely devastated many diverse populations of Native Americans, who had no natural resistance to many European diseases. It has been estimated that up to 90% of Native Americans died from infectious diseases after the arrival of Europeans, making conquest of the New World a foregone conclusion. Emerging and re-emerging diseases. The distribution of a particular disease is dynamic. Changes in the environment, the pathogen, or the host population can dramatically impact the spread of a disease. According to the World Health Organization, WHO, an emerging disease, figure 22. 22 is one that has appeared in a population for the first time, or that may have existed previously, but is rapidly increasing in incidence or geographic range. This definition also includes re-emerging diseases that were previously under control. Approximately 75% of recently emerging infectious diseases affecting humans are zoonotic diseases. Zoonoses are diseases that primarily infect animals but can be transmitted to humans. Some are of viral origin and some are of bacterial origin. Brucellosis is an example of a prokaryotic zoonosis that is re-emerging in some regions, and necrotizing fasciitis, commonly known as flesh-eating bacteria, has been increasing in virulence for the last 80 years for unknown reasons. Emerging or re-emerging bacterial diseases are shown on a world map. Multi-drug-resistant tuberculosis is emerging in North America, Europe, North Africa, and Asia. Vancomycin-resistant Staphylococcus aureus and E. coli 0157:H7 are emerging in North America and East Asia. Lyme disease is spreading in North America. Cholera is emerging in Africa and South America. Diphtheria and typhoid fever are re-emerging in Asia. Figure 22. 22 emerging diseases. The map shows regions where bacterial diseases are emerging or re-emerging. Credit, modification of work by NIH. Some of the present emerging diseases are not actually new, but are diseases that were catastrophic in the past. Figure 22. 23. They devastated populations and became dormant for a while, just to come back, sometimes more virulent than before, as was the case with bubonic plague. Other diseases, like tuberculosis, were never eradicated, but were under control in some regions of the world until coming back, mostly in urban centers with high concentrations of immunocompromised people. Who has identified certain diseases whose worldwide reemergence should be monitored? Among these are 3 viral diseases, dengue fever, yellow fever, and Zika, and 3 bacterial diseases, diphtheria, cholera, and bubonic plague. The war against infectious diseases has no foreseeable end. Part A shows the red, bullseye-shaped rash of a person infected with Borrelia. Part B shows a micrograph of Borrelia, which look like tiny corkscrews. Part C shows the life cycle of the bacteria, which begins when Borrelia infect a tick egg. The egg hatches into larva, which feeds on a mouse, then into a nymph, which also feeds on a mouse. The nymph feeds again, this time on a deer or sometimes a human. Figure 22. 23 Lyme disease. Lyme disease often, but not always, results in a characteristic bullseye rash. The disease is caused by a B gram-negative spirochete bacterium of the genus Borrelia. The bacteria, C, infect ticks, which in turn infect mice. Deer are the preferred secondary host, but the ticks also may feed on humans. Untreated, the disease causes chronic disorders in the nervous system, eyes, joints, and heart. The disease is named after Lyme, Connecticut, where an outbreak occurred in 1995 and has subsequently spread. The disease is not new, however. Genetic evidence suggests that Ötzi the Iceman, a 5,300-year-old mummy found in the Alps, was infected with Borrelia. Credit A. James Gatheny, CDC, Credit B. CDC. Scalebar data from Matt Russell. Foodborne diseases. Prokaryotes are everywhere. They readily colonize the surface of any type of material, and food is not an exception. Most of the time, prokaryotes colonize food and food processing equipment in the form of a biofilm, as we have discussed earlier. Outbreaks of bacterial infection related to food consumption are common. A foodborne disease, commonly called food poisoning, is an illness resulting from the consumption of the pathogenic bacteria, viruses, or other parasites that contaminate food. Although the United States has one of the safest food supplies in the world, the U.S. Centers for Disease Control and Prevention, CDC, has reported that 76 million people get sick, more than 300,000 are hospitalized, and 5,000 Americans die each year from foodborne illness. The characteristics of foodborne illnesses have changed over time. In the past, it was relatively common to hear about sporadic cases of botulism, the potentially fatal disease produced by a toxin from the anaerobic bacterium Clostridium botulinum. Some of the most common sources for this bacterium were non-acidic canned foods, homemade pickles, and processed meat and sausages. The can, jar, or package created a suitable anaerobic environment where Clostridium could grow. Proper sterilization and canning procedures have reduced the incidence of this disease. While people may tend to think of foodborne illnesses as associated with animal-based foods, most cases are now linked to produce. There have been serious, produce-related outbreaks associated with raw spinach in the United States and with vegetable sprouts in Germany, and these types of outbreaks have become more common. The raw spinach outbreak in 2006 was produced by the bacterium E. coli serotype O157, H7. A serotype is a strain of bacteria that carries a set of similar antigens on its cell surface, and there are often many different serotypes of a bacterial species. Most E. coli are not particularly dangerous to humans, but serotype O157, H7 can cause bloody diarrhea and is potentially fatal. All types of food can potentially be contaminated with bacteria. Recent outbreaks of salmonella, reported by the CDC, occurred in foods as diverse as peanut butter, alfalfa sprouts, and eggs. A deadly outbreak in Germany in 2010 was caused by E. coli contamination of vegetable sprouts, figure 22, 24. The strain that caused the outbreak was found to be a new serotype not previously involved in other outbreaks, which indicates that E. coli is continuously evolving. Outbreaks of listeriosis, due to contamination of meats, raw cheeses, and frozen or fresh vegetables with Listeria monocytogenes, are becoming more frequent. Photo A shows round green seeds with stems sprouting from them. Micrograph B shows rod-shaped bacteria about 2 microns in length. Sketch C shows normal, disc-shaped red blood cells on the left. On the right, many of the red blood cells are incomplete or fragmented. Figure 22, 24 foodborne pathogens. A. Vegetable sprouts grown at an organic farm were the cause of an B. E. coli outbreak that killed 32 people and sickened 3,800 in Germany in 2011. The strain responsible, E. coli O104. H4 produces Shiga toxin, a substance that inhibits protein synthesis in the host cell. The toxin, C, destroys red blood cells resulting in bloody diarrhea. Deformed red blood cells clog the capillaries of the kidney, which can lead to kidney failure, as happened to 845 patients in the 2011 outbreak. Kidney failure is usually reversible, but some patients experience kidney problems years later. Credit C, NIDDK, NIH. Biofilms and disease. Recall that biofilms are microbial communities that are very difficult to destroy. They are responsible for diseases such as Legionnaire's disease, otitis media, ear infections, and various infections in patients with cystic fibrosis. They produce dental plaque and colonize catheters, prostheses, transcutaneous and orthopedic devices, contact lenses, and internal devices such as pacemakers. They also form in open wounds and burn tissue. In healthcare environments, biofilms grow on hemodialysis machines, mechanical ventilators, shunts, and other medical equipment. In fact, 65% of all infections acquired in the hospital, nosocomial infections, are attributed to biofilms. Biofilms are also related to diseases contracted from food because they colonize the surfaces of vegetable leaves and meat, as well as food processing equipment that isn't adequately cleaned. Biofilm infections develop gradually and may not cause immediate symptoms. They are rarely resolved by host defense mechanisms. Once an infection by a biofilm is established, it is very difficult to eradicate because biofilms tend to be resistant to most methods used to control microbial growth, including antibiotics. The matrix that attaches the cells to a substrate and to another protects the cells from antibiotics or drugs. In addition, since biofilms grow slowly, they are less responsive to agents that interfere with cell growth. It has been reported that biofilms can resist up to 1,000 times the antibiotic concentrations used to kill the same bacteria when they are free-living or planktonic. An antibiotic dose that large would harm the patient. Therefore, scientists are working on new ways to get rid of biofilms. Antibiotics. Are we facing a crisis? The word antibiotic comes from the Greek anti meaning against and bios meaning life. An antibiotic is a chemical, produced either by microbes or synthetically, that is hostile to or prevents the growth of other organisms. Today's media often address concerns about an antibiotic crisis. Are the antibiotics that easily treated bacterial infections in the past becoming obsolete? Are there new superbugs, bacteria that have evolved to become more resistant to our arsenal of antibiotics? Is this the beginning of the end of antibiotics? All these questions challenge the healthcare community. One of the main causes of antibiotic resistance in bacteria is overexposure to antibiotics. The imprudent and excessive use of antibiotics has resulted in the natural selection of resistant forms of bacteria. The antibiotic kills most of the infecting bacteria, and therefore only the resistant forms remain. These resistant forms reproduce, resulting in an increase in the proportion of resistant forms over non-resistant ones. In addition to transmission of resistance genes to progeny, lateral transfer of resistance genes on plasmids can rapidly spread these genes through a bacterial population. A major misuse of antibiotics is in patients with viral infections like colds or the flu, against which antibiotics are useless. Another problem is the excessive use of antibiotics in livestock. The routine use of antibiotics in animal feed promotes bacterial resistance as well. In the United States, 70% of the antibiotics produced are fed to animals. These antibiotics are given to livestock in low doses, which maximize the probability of resistance developing, and these resistant bacteria are readily transferred to humans. Link to learning. Watch a recent news report on the problem of routine antibiotic administration to livestock and antibiotic-resistant bacteria. One of the superbugs, MRSA. The imprudent use of antibiotics has paved the way for the expansion of resistant bacterial populations. For example, Staphylococcus aureus, often called staph, is a common bacterium that can live in the human body and is usually easily treated with antibiotics. However, a very dangerous strain, methicillin-resistant Staphylococcus aureus, MRSA, has made the news over the past few years. Figure 22. 25. This strain is resistant to many commonly used antibiotics, including methicillin, amoxicillin, penicillin, and oxacillin. MRSA can cause infections of the skin, but it can also infect the bloodstream, lungs, urinary tract, or sites of injury. While MRSA infections are common among people in healthcare facilities, they have also appeared in healthy people who haven't been hospitalized, but who live or work in tight populations, like military personnel and prisoners. Researchers have expressed concern about the way this latter source of MRSA targets a much younger population than those residing in care facilities. The Journal of the American Medical Association reported that, among MRSA-afflicted persons in healthcare facilities, the average age is 68, whereas people with community-associated MRSA, CAMRSA, have an average age of 23. 4. The micrograph shows clusters of round bacteria clinging to a surface. Each bacterium is about 0.4 microns across. Figure 22, 25 MRSA. This scanning electron micrograph shows methicillin-resistant Staphylococcus aureus bacteria, commonly known as MRSA. S. aureus is not always pathogenic, but can cause diseases such as food poisoning and skin and respiratory infections. Credit, modification of work by Janice Haney-Carr, scale bar data from Matt Russell. In summary, the medical community is facing an antibiotic crisis. Some scientists believe that, after years of being protected from bacterial infections by antibiotics, we may be returning to a time in which a simple bacterial infection could again devastate the human population. Researchers are developing new antibiotics, but it takes many years of research and clinical trials, plus financial investments in the millions of dollars, to generate an effective and approved drug. Career connection. Epidemiologist. Epidemiology is the study of the occurrence, distribution, and determinants of health and disease in a population. It is, therefore, part of public health. An epidemiologist studies the frequency and distribution of diseases within human populations and environments. Epidemiologists collect data about a particular disease and track its spread to identify the original mode of transmission. They sometimes work in close collaboration with historians to try to understand the way a disease evolved geographically and over time, tracking the natural history of pathogens. They gather information from clinical records, patient interviews, surveillance, and any other available means. That information is used to develop strategies, such as vaccinations, figure 22, 26, and design public health policies to reduce the incidence of a disease or to prevent its spread. Epidemiologists also conduct rapid investigations in case of an outbreak to recommend immediate measures to control it. An epidemiologist has a bachelor's degree plus a master's degree in public health (MPH). Many epidemiologists are also physicians and have an MD or DO degree, or they have a Ph.D. in an associated field such as biology or microbiology. This photo shows syringes, adhesive bandages, and alcohol swabs. Figure 22, 26 vaccination. Vaccinations can slow the spread of communicable diseases. Credit, modification of work by Daniel Paquet. Learning objectives. By the end of this section, you will be able to do the following. Explain the need for nitrogen fixation and how it is accomplished. Describe the beneficial effects of bacteria that colonize our skin and digestive tracts. Identify prokaryotes used during the processing of food. Describe the use of prokaryotes in bioremediation. Fortunately, only a few species of prokaryotes are pathogenic. Prokaryotes also interact with humans and other organisms in a number of ways that are beneficial. For example, prokaryotes are major participants in the carbon and nitrogen cycles. They produce or process nutrients in the digestive tracts of humans and other animals. Prokaryotes are used in the production of some human foods and also have been recruited for the degradation of hazardous materials. In fact, our life would not be possible without prokaryotes. Cooperation between bacteria and eukaryotes. Nitrogen fixation. Nitrogen is a very important element to living things because it is part of nucleotides and amino acids that are the building blocks of nucleic acids and proteins, respectively. Nitrogen is usually the most limiting element in terrestrial ecosystems, with atmospheric nitrogen, N2, providing the largest pool of available nitrogen. However, eukaryotes cannot use atmospheric gaseous nitrogen to synthesize macromolecules. Fortunately, nitrogen can be fixed, meaning it is converted into a more accessible form, ammonia, NH3, either biologically or abiotically. Abiotic nitrogen fixation occurs as a result of physical processes such as lightning or by industrial processes. Biological nitrogen fixation, BNF, is exclusively carried out by prokaryotes, soil bacteria, cyanobacteria, and Frankia spp. Filamentous bacteria interacting with actinorhizal plants such as alder, bayberry, and sweet fern. After photosynthesis, BNF is the most important biological process on Earth. The overall nitrogen fixation equation below represents a series of redox reactions. Pi stands for inorganic phosphate. N2 plus 16 ATP plus 8 E minus plus 8H plus right arrow 2NH3 plus 16 ADP plus 16 pi plus H2. The total fixed nitrogen through BNF is about 100 to 180 million metric tons per year, which contributes about 65% of the nitrogen used in agriculture. Cyanobacteria are the most important nitrogen fixers in aquatic environments. In soil, members of the genera Clostridium and Azotobacter are examples of free-living, nitrogen-fixing bacteria. Other bacteria live symbiotically with legume plants, providing the most important source of fixed nitrogen. Symbionts may fix more nitrogen in soils than free-living organisms by a factor of 10. Soil bacteria, collectively called rhizobia, are able to symbiotically interact with legumes to form nodules, specialized structures where nitrogen fixation occurs. Figure 22. 27. Nitrogenase, the enzyme that fixes nitrogen, is inactivated by oxygen, so the nodule provides an oxygen-free area for nitrogen fixation to take place. The oxygen is sequestered by a form of plant hemoglobin called leg hemoglobin, which protects the nitrogenase, but releases enough oxygen to support respiratory activity. Symbiotic nitrogen fixation provides a natural and inexpensive plant fertilizer. It reduces atmospheric nitrogen to ammonia, which is easily usable by plants. The use of legumes is an excellent alternative to chemical fertilization and is of special interest to sustainable agriculture, which seeks to minimize the use of chemicals and conserve natural resources. Through symbiotic nitrogen fixation, the plant benefits from using an endless source of nitrogen, the atmosphere. The bacteria benefit from using photosynthates, carbohydrates produced during photosynthesis, from the plant and having a protected niche. In addition, the soil benefits from being naturally fertilized. Therefore, the use of rhizobia as biofertilizers is a sustainable practice. Why are legumes so important? Some, like soybeans, are key sources of agricultural protein. Some of the most important legumes consumed by humans are soybeans, peanuts, peas, chickpeas, and beans. Other legumes, such as alfalfa, are used to feed cattle. This photo shows a legume root, which is thin and yellow with nodules sticking out of it. Figure 22. 27 nitrogen fixation nodules on soybean roots. Soybean glycine max is a legume that interacts symbiotically with the soil bacterium Bradyrhizobium japonicum to form specialized structures on the roots called nodules where nitrogen fixation occurs. Credit: USDA. Everyday connection. Microbes on the human body. The commensal bacteria that inhabit our skin and gastrointestinal tract do a host of good things for us. They protect us from pathogens, help us digest our food, and produce some of our vitamins and other nutrients. These activities have been known for a long time. More recently, scientists have gathered evidence that these bacteria may also help regulate our moods, influence our activity levels, and even help control weight by affecting our food choices and absorption patterns. The Human Microbiome Project has begun the process of cataloging our normal bacteria and Archaea so we can better understand these functions. A particularly fascinating example of our normal flora relates to our digestive systems. People who take high doses of antibiotics tend to lose many of their normal gut bacteria, allowing a naturally antibiotic-resistant species called Clostridium difficile to overgrow and cause severe gastric problems, especially chronic diarrhea. Figure 22. 28. Obviously, trying to treat this problem with antibiotics only makes it worse. However, it has been successfully treated by giving the patients fecal transplants from healthy donors to reestablish the normal intestinal microbial community. Clinical trials are underway to ensure the safety and effectiveness of this technique. Micrograph shows small clusters of white rod-shaped bacteria against a dark background. Figure 22. 28 Clostridium difficile. This scanning electron micrograph shows Clostridium difficile, a gram-positive, rod-shaped bacterium that causes severe diarrhea. Infection commonly occurs after the normal gut fauna are eradicated by antibiotics, and in the hospital can be deadly to seriously ill patients. Credit: modification of work by CDC, HHS. Scalebar data from Matt Russell. Scientists are also discovering that the absence of certain key microbes from our intestinal tract may set us up for a variety of problems. This seems to be particularly true regarding the appropriate functioning of the immune system. There are intriguing findings that suggest that the absence of these microbes is an important contributor to the development of allergies and some autoimmune disorders. Research is currently underway to test whether adding certain microbes to our internal ecosystem may help in the treatment of these problems, as well as in treating some forms of autism. Early biotechnology. Cheese, bread, wine, beer, and yogurt. According to the United Nations Convention on Biological Diversity, biotechnology is any technological application that uses biological systems, living organisms, or derivatives thereof to make or modify products or processes for specific use. 5. The concept of specific use involves some sort of commercial application. Genetic engineering, artificial selection, antibiotic production, and cell culture are current topics of study in biotechnology and will be described in later chapters. However, humans were using prokaryotes before the term biotechnology was even coined. Some of the products of this early biotechnology, as familiar as cheese and yogurt, employ bacteria, while other microbes, such as yeast, a fungus, figure 22. 29, are used to produce bread, wine, and beer. The photo collage shows cheese, yogurt, wine, and beer and bread. Figure 22. 29, some foods produced by microorganisms. Some of the products derived from the use of prokaryotes in early biotechnology include a cheese, B, yogurt, C, beer and bread, and D, wine. Credit bread, modification of work by F. Rodrigo/Wikimedia Commons. Credit wine, modification of work by John Sullivan. Credit beer and bread, modification of work by Chris Miller. Credit yogurt, modification of work by John Sullivan. Cheese production began around 4,000 to 7,000 years ago when humans began to breed animals and process their milk. Fermentation in this case preserves nutrients. Milk will spoil relatively quickly, but when processed as cheese, it is more stable. As for beer, the oldest records of brewing are about 6,000 years old and were an integral part of the Sumerian culture. Evidence indicates that the Sumerians discovered fermentation by chance. Wine has been produced for about 4,500 years, and evidence suggests that cultured milk products, like yogurt, have existed for at least 4,000 years. Using prokaryotes to clean up our planet, bioremediation. Microbial bioremediation is the use of prokaryotes, or microbial metabolism, to remove pollutants. Bioremediation has been used to remove agricultural chemicals, e.g., pesticides, fertilizers, that leach from soil into groundwater and the subsurface. Certain toxic metals and oxides, such as selenium and arsenic compounds, can also be removed from water by bioremediation. The reduction of CO4-2 to CO3-2 and to C0, metallic selenium, is a method used to remove selenium ions from water. Mercury, Hg, is an example of a toxic metal that can be removed from an environment by bioremediation. As an active ingredient of some pesticides, mercury is used in industry and is also a byproduct of certain processes, such as battery production. Methylmercury is usually present in very low concentrations in natural environments, but it is highly toxic because it accumulates in living tissues. Several species of bacteria can carry out the biotransformation of toxic mercury into non-toxic forms. These bacteria, such as Pseudomonas aeruginosa, can convert Hg+2 into Hg0, which is non-toxic to humans. One of the most useful and interesting examples of the use of prokaryotes for bioremediation purposes is the cleanup of oil spills. The significance of prokaryotes to petroleum bioremediation has been demonstrated in several oil spills in recent years, such as the Exxon Valdez spill in Alaska, 1989, figure 22. 30, the Prestige oil spill in Spain, 2002, the spill into the Mediterranean from a Lebanon power plant, 2006, and more recently, the BP oil spill in the Gulf of Mexico, 2010. In the case of oil spills in the ocean, ongoing natural bioremediation tends to occur, since there are oil-consuming bacteria in the ocean prior to the spill. In addition to these naturally occurring oil-degrading bacteria, humans select and engineer bacteria that possess the same capability with increased efficacy and spectrum of hydrocarbon compounds that can be processed. Bioremediation is enhanced by the addition of inorganic nutrients that help bacteria to grow. Some hydrocarbon-degrading bacteria feed on hydrocarbons in the oil droplet, breaking down the hydrocarbons into smaller subunits. Some species, such as Alcanivorax borcumensis, produce surfactants that solubilize the oil, making it soluble in water, whereas other bacteria degrade the oil into carbon dioxide. Under ideal conditions, it has been reported that up to 80% of the non-volatile components in oil can be degraded within 1 year of the spill. Other oil fractions, containing aromatic and highly branched hydrocarbon chains, are more difficult to remove and remain in the environment for longer periods of time. Part A, this photo shows 2 men in yellow ranger hosing off oil-drenched rocks on a seashore. Part B, this photo shows an oil-drenched bird sitting in oily water. Figure 22. 30 prokaryotes and bioremediation. A. Cleaning up oil after the Exxon Valdez spill in Alaska. Workers hosed oil from beaches and then used a floating boom to corral the oil, which was finally skimmed from the water surface. Some species of bacteria are able to solubilize and degrade the oil. B. One of the most catastrophic consequences of oil spills is the damage to fauna. Credit, a modification of work by NOAA, credit B. Modification of work by Giolubinkov, NGO, saving Taman.